Okay, hello everybody. Let's continue on with vision. I know this is sort of the eternal sense, but you'll see how the other ones will come by much quicker. So anyway, in this last part of vision, I'd like to address two fundamental questions to end this sense for once. How is it that we see color, and also what happens to visual processing once these messages or neural impulses go through the optic nerve? Meaning, how is it that we organize what we see, and how is it that the brain makes sense of what we see? But going back to the first question that I posed, let's remember that although the spectrum of visible light. Remember visible light, the variety of wavelengths that are detectable by us, by humans. Although it's only a minimal portion of the entire electromagnetic spectrum, we can sort of pat ourselves on the back too, because our eyes can discriminate between or distinguish among 7.5 million colors. And that's pretty amazing when you think about it. 7.5 million colors. So the pressing question is, however, how is it that we can perceive color? Because remember, there is no color around us. Color is a perceptual construct in a way, or it is a perceptual construct. In fact, the color we perceive of a particular object, let's say a red tomato, the tomato is everything but red. In fact, the wavelength that bounces off the tomato is what goes into our retina and what we interpret as red. So after how we organize these wavelengths that are entering through our cornea. But anyway, there are two possible theories that explain the colors we interpret from around us. And these are the Young-Helmholtz trichromatic theory and opponent process theory, and I'll proceed to explain these. It never hurts to review, with so many terms, the basics. So I'll repeat. With cones, we accomplish color vision. Remember, cones are located in the fovea, area of the retina that we've labeled in the screencast as VIP, because it requires a lot of light. This area, it only houses cones. They're quite they're quite royalty, and therefore they provide us with a great level of detail and color. So chromatic vision is accomplished with or through cones. Achromatic vision without color, is accomplished by our rods. Rods help us define or perceive vision, sense and perceive vision in terms of our peripheral visual field. Therefore, because that's their responsibility, our periphery of vision, they require less light, less energy. However, the details and colors are not accomplished in our peripheral vision. So when we talk about color vision, we're talking about cones. The first theory is perhaps the most simple one. It's a Young-Helmholtz trichromatic theory, and it proposes something quite basic. And it says that we have basically three types of cones, and each of these cones specializes in a particular light wave. Because remember that I try to avoid saying that each of these cones specializes in a color, at least at this point, to avoid that confusion that color is something that exists. Remember, color is a perceptual construct. It's what we basically, colloquially, is what we do with these different light waves. So we perceive them, we interpret them as color. If you're looking at a red tomato, in fact, that tomato is all but red, because red is what's bouncing off the surface of that tomato and entering our cornea, and that light wave that we receive from that we receive bouncing off that tomato is what we perceive as red. So remember, color does not exist. It's our interpretation of that particular energy that we are inputting through our eyes. Just to leave that very clear. So we have three types of cones that specialize in different waves. In those waves that correspond to bluish colors, we have cones for that. We have cones that specialize in waves that correspond to the greenish tones of color, and we have another type of cones that are responsible for processing light energy, that light waves that correspond to the reddish tonalities of color. So again, each type of cone sort of is responsible for one of the primary colors of light, or the wavelength that corresponds to that perceived hue, or what we perceive as color. Again, we have cones that detect the different color blues. We have cones that detect the different colors of green, and we have cones that detect the different colors of red. This means that it's quite big, because it means that each of those 7 million colors that we can detect, because we can differentiate among them. I mentioned that previously, sorry. Of the possibilities within the spectrum of visible light is a result of a combination of these three primary colors. So very simple. We have three different type of cones. They process their corresponding light waves and what we perceive as colors. And all this variety of color within the visible spectrum, remember what humans can perceive as color or detect, is a combination of these three. Now, this research continues to support Young-Helmholtz trichromatic theory. I hate pronouncing that title for some reason. But there were some phenomena that this Young-Helmholtz trichromatic theory could not explain for. And these things or phenomena are color blindness and after images. Because basically what happens is this. I'll explain what after images are in a minute, but with color blindness, by the way, most colorblind people are what gender or what sex? Male. Yes. So usually it's due to deficiency in the cones, or it's either deficiency in the cones, or they basically don't have the cones that correspond to the particular color that they're deficient with. Okay, so let's go back to opponent process theory. So opponent process theory, I have to leave it very clear that they're not sort of shunning Young-Helmholtz trichromatic theory. They are basically saying that there are additional steps through which we process what we perceive as color. Because think about this, usually, and this is important to know, usually colorblind people are, and it's usually a term that can be confusing, because colorblind does not mean, usually it's very rare for somebody to have achromatic vision completely. Usually people that are colorblind, mostly males, remember, either have trouble processing, they can't process red and green, or they cannot process blue and yellow. Those are the most common conditions of color blindness. Again, they cannot process two colors. Either it's red and green or blue and yellow. So think about this. If we go back to Young-Helmholtz trichromatic theory, how is it that if you cannot process, if let's say there's a problem with your cones that correspond to red and green, how is it that colorblind people process yellow? And so again, I hope it's self-explanatory that this theory was insufficient to explain color blindness. So then opponent process theory adds to that process and says that that there are three different types of cones, but that the processing of these light waves, requires or goes through additional steps. And that in these additional steps, these cones are paired. Green cones that process green work with their red companions. Cones that process blue work side to side with their yellow companions. And cones that work with black or no light work with white light, which is a combination of the absence of light with those that process all lights together, which leads to white. So again, opponent process theory says that these cones work in pairs. They work together hand in hand. The other important thing, I mean, think about the name opponent, and they say that, think about all these pairs, right? Each pair, they have a sort of a dysfunctional relationship with each other. Think about it as a dysfunctional marriage in which they can't live without each other, but they can live with each other too. When green is up on stage, right, processing what corresponds to their wavelengths, red is inhibited. Green is stimulated at the expense. That's why I put the seesaw as a picture. That if green is working, red cannot work. It needs green, but it can't work while green is working, and vice versa. And the same with blue. If we're processing blue, then we cannot process yellow. Yellow cones that process yellow, although there are pairs with these cones that process blue, have to be completely inhibited, and so forth. So, and this sort of is compatible with color blindness. Again, mentioned previously, those who suffer from color blindness, the most common condition are people that cannot process green and red, another condition is when they cannot process blue or yellow, meaning that that these cones work together, are intrinsically, I forget what I'm going to say, but they're tied together in a very specific and unique way that they depend on each other. If you don't have one, you don't have the other. But the work of one comes at the expense of the work of other, and I'm going to explain that in a minute too. So again, based on this processing, is that that they sort of say this is in addition to this primary classification that young Helmholtz trichromatic theory proposes. They also add, they also work in pairs too, and these are the pairs that work together. Opponent process theory explains after images, and I'd like to invite you to participate in this exercise so you know what I'm talking about. These pairs of cones work together, but they never work at the same time. Sort of when you're processing green, your red is inhibited. So opponent meaning that they work against each other, with and against each other. When what happens is when we stare at an image, we fatigue those cones that are processing the green, and basically when we find a neutral, space, then their opponent will come back full force. Meaning, okay, you didn't let me work for all this time while you were staring at that stupid flag? Well, I mean stupid just because it wasn't the right one. I mean, don't misinterpret it, please. So while you were staring at that flag, you left me here inhibited? Okay, well, now we have this white surface. Now I am coming full force. And this is basically what happens with each pair of working cones that are processing color. And this is basically, I know it's very rudimentary, I'm not a specialist in this, but it's as far as you need to go in terms of how we process color vision. Here we go. I am going to present to you a flag, an image, regardless of whatever image it is, and please stare at it completely. Don't move your eyes or your vision to another object or anything, because then you just don't see the results or you don't get the after image. But I'd like you, many of you I'm sure have done this before, try to you're going to stare at this image for approximately 30 seconds, and I will remove it from the screencast. Okay, ready? Very easy. Just focus on the image, try not to blink, and just look at it, stare at it for approximately 30 seconds. Here we go. Now stare at that. Okay, so hopefully, and you can repeat this exercise on your own, but hopefully what you saw was that from a green, black, and yellow, green, black flag, you saw an after image on the white surface as the colors of the corresponding colors of the American flag, the flag of the United States of America. So now, how does this after image explain opponent process theory? Well, again, remember that we said that opponent process theory establishes that the second question, the first question was how we process color. Then finally, how does the brain make sense of everything that we see? Sort of the entire visual processing in a nutshell. So we've studied the eye. We've talked again and again about how light waves are sensed, beginning as they pass through our cornea and the anatomy of the eye, they project onto the surface of the retina. And remember, our vision is actually quite imperfect. First of all, the image that is projected on the retina is upside down. We also, considering the blind spots, we should see actually with these two, black holes sort of following us everywhere we see. But our brain says, oh, okay, no, I don't like these two sort of holes, circles in my way. So basically our brain is constantly filling up those gaps. We talked about that. So the reason why I'm bringing this up again is because our brain participates in this process as much as our eyes do. And it basically sort of cleans what we sense by perceiving it, organizing it, interpreting it in a very convenient way for us. Being one that we don't see those blind spots unless we do some of those exercises practiced in the previous screencast. So the work of David Schubel, I think, and Wiesel, I think is German. They won actually a Nobel Prize. It's in your textbook for identifying what they denominated as feature detectors. And what happens is this. Believe it or not, so once the cones and the rods react chemically, and then they go through the other two layers of the retina, the bipolar cells and the ganglion receptors, then they go through the optic nerve and they travel to our brain, where all this information that we've that we've sensed from our environment is going to be processed. Of course, they make that first stop. Remember, the thalamus acts as sort of the pit stop of most senses, except for smell doesn't go through the thalamus, and then it's taken to, these types of signals correspond to the visual cortex located in our occipital lobes, in this area right here. Now here, important occurs, and again, as I was mentioning, this is where our vision is actually, okay, so with this image, we can see that there are areas, feature detectors are, again, cells that specialize in particular images, particular aspects of an image, or such as angles, lines, but also as objects. We have areas specializing in the recognition of faces. Those who suffer from prosopagnosia have this area damaged or genetically predetermined to dysfunction in this area of the brain. But we have areas, it's quite fascinating, we have areas in the brain that specialize in houses. And prosopagnosia patients can, in fact, process houses. They can see a house and differentiate one from another. They just can't do the face recognition. And the reason why I brought this up, too, is because this is another example of how specialized these cells can be. When we look at a face, when we see somebody, even if we're familiar with that person or not, I repeat, we're processing the eyes, we're processing the mouth, we're processing a particular shape of mouth, eyes, everything, hair, and everything. And then we have sort of this internal album of possible face combinations. And I know it sounds sort of out of whack, but it is the case that we compare it to. Okay, so this person falls in within these possibilities of facial combinations of eye, mouth, all these features. And then we compare it to these, and then we say, oh, this person is my grandmother. Oh, this person is my mother. And again, something difficult to believe because it's something we take for granted and we do basically automatically without even thinking as much as we think we do. So that's all for visual information processing. You have more information in your textbook, and I'd be glad to answer any questions if you have any. Thank you. A little counterintuitive, and it might be in fact hard to believe that we do all this processing for every single thing that we see during every single minute, second of our lives. But basically, what Hubel and Wiesel found were, I forgot their names, was that we have cells that sort of specialize in particular features of every single object that we see. So basically, our brains do the following questioning as they watch or see something. Is it going to move? So we have particular cells that are, by the way, complex cells, because motion is sort of complicated than just a single line or a couple of lines. But then again, we have cells that specialize in motion, so the brain asks, is this object moving? Is it going to move? Is it not moving? Is it static? Will it stay static? And we have cells that specialize in that part of the image. Then we also have other cells that participate or process form. They specialize in that department. And then we have other cells that specialize in depth. And we have others that specialize in color, which are, as you might remember, these are the cones. And we already talked about the different theories that explain this particular specialization. The point here being is that then once we detect that this is what we call parallel processing, we're doing all of these cells are working independently. And then later, they are going to bring this all together. They're working at the same time and later bring that image that we are seeing. Now again, it's hard to believe that we do this. And to avoid some confusion, I'm going to put this blind side. I'll explain why I had that there. But this parallel processing consists of specializing in different features of what we're seeing, color, form, depth. In fact, we have some cells that specialize in lines and angles, and all of this is happening at the same time, simultaneously, and then later all this information is going to be put together. So our brain is basically breaking down everything that we see to its very basic elements to then build it up again. Which is something quite fascinating. It occurs so instantaneously, and yet, and again, it's hard to believe that we do this. And so just to recap, and this is found in your textbook, by the way, we begin with what we see, right? What is in our visual field, and then that leads to certain light waves that are going to enter our system through our eyes, and then later projected on the retina. Here's where the processing occurs when what I just explained again and again, the receptor cells or photoreceptors, the rods and the cones are processing these light waves, then activate the bipolar cells and the ganglion cells, and then they travel through the optic nerve. They are going to travel these neural signals because transduction has occurred in the retina. Now that its electrochemical energy enters our occipital lobe, specifically the visual cortex, and within this visual cortex we have different areas where there are different cells that are specialized in one particular basic feature of the image that we are seeing. So we have cells for edges, we have cells for lines, we have cells for angles. As I was saying, very basic features, and our brain breaks it down and says, okay, you guys process this, you guys process that, and then finally, the parallel processing consists of, okay, each team, do you have your work done? Okay, now let's bring it all together, and they provide us with an information. In this case, so we see the tiger, we break it down to its basic elements, and then we put them all together, and then it provides us, once we see this image, we have particular cells that in fact tell us, oh, okay, well, this tiger looks like as if it were going to jump or strike or attack or something. And all of this is processed instantaneously, basically, in the matter of, seconds. I would say even less than that. And then finally, an important part is that recognition. This is the final part of the visual processing, visual information processing, and believe it or not, too, automatically, our brain doesn't say, well, automatically, oh, this is a tiger that is looking this way and is about to move. What we do is with everything we see, even if we are familiar with it, we compare, okay, well, now that I have this image all solved, and I have it all built, right, breaking it down and then building it up again, basically, with my feature detectors, with these cells that specialize in very particular features of an image, once, and the parallel processing has occurred, then I say, okay, well, this is not a cat, this is not a dog, this is not an elephant, this is not similar. We have sort of these very sort of similar, well, I know an elephant or a dog are not very similar to a tiger, but we have these schemas, let's put it that way, that we compare. Is it this? Is it this? No, it's not this. Oh, it's this. We are basically recognizing the image from something that we have stored based on our experiences and memories. I'd like to come back here to the visual information processing and explain that face recognition. Remember the condition of prosopagnosia, people who have no problem seeing, this is another, in addition to blind sight, also demonstrates the complexity of visual processing as a whole. People that suffer from prosopagnosia, if you remember from that 60-minute video, their vision is intact. They can see things. They can recognize houses because, in fact, I believe we have this figure right here. I forget where it was. Oh, we spoke about blind sight in our biopsych unit. And the reason why it comes up again is because blind sight patients provide support to this theory that, plenty of other evidence has been found, but blind sight, patients of blind sight or who experience blind sight support and provide evidence to this fact that our brain our brain is very complex in the way it processes light energy, in that perhaps we are consciously not seeing something because, perhaps this patient had a stroke that affected a particular area that allows that person to not see. Or there's a particular temporary condition that doesn't allow that person to see and therefore to recognize his or her surroundings consciously. Yet, our brain, perhaps other areas, haven't been damaged by this stroke. And this is the reason why, although a person might not report to see something, they will act because, again, these other areas of our cortex, of our visual cortex, are processing the information regardless of whether we are conscious about it or not. So blind sight patients, again, give us an insight, no pun intended, to the complexity of the visual processing. It's not just one way or one channel through which we process these light waves. Instead, it's complex. Once these neural impulses are sent to the brain, you might not be consciously aware, but your brain is working with the information, with the energy it receives from its environment. This is why blind sight, it comes up in this part of the unit as well.